Welcome to the Crop Physiology course. In this presentation we will address the first part of Unit 1, which includes crop development and phenology. We will start with a brief analysis of the importance of phenological knowledge and how we use it by means of different scales for describing the life cycle of the main grain crop species. We will talk about phasic development, that is, the different stages we use to describe a growth cycle, and morphological development, that is, the generation of the phenotype. We will introduce the concept of plastochron associated to meristem's activity and phyllochron associated to leaf expansion. The importance of phenology is very clear when we analyze grain yield response of any species to its cycle duration. The example in this figure corresponds to rice grown under rain-fed farming in India, and its grain yield response is characterized by the dashed line. We can observe a curvilinear, optimum type response where grain yield is maximized at a given range of cycle duration. To the left of this range, grain yield is limited by crop capacity to capture resources, for instance due to incomplete ground cover. To the right of this range, cycle durations are excessively long and grain yield is limited because crops are exposed to less favorable conditions during critical periods for grain yield determination. The described trend is verified in all annual species we will address in the course. In this example for soybean, we can observe that there are differences in documented response depending upon the environment. The graph on the left corresponds to a high-latitude environment, representative, for example, of Iowa in the United States. In this environment, grain yield is maximized with cycle durations shorter than 150 days, which are obtained with soybean maturity groups between 2 and 4. The graph on the right corresponds to a mid-latitude environment, representative, for example, of Gainesville, Florida. In this environment, grain yield is maximized with cycle durations of at least 130 days, which are obtained with maturity groups between 6 and 9. Evidently, knowledge of growth cycle and its variation across environments is crucial for adapting crop management at each site. The first approach to the study of phenology was of a qualitative type, based on the description of visible, conspicuous changes produced by plants along their life cycle. In this schematic representation, we have a simplified version of representative stages of an annual crop. These are the vegetative phase, the reproductive phase, and the maturation or grain filling phase. Different organs are initiated and start growing in each phase. During this differentiation process, some traits are easy to observe and constitute the so-called external phenotype. Examples of this group are germination, leaf expansion, flowering, fruit growth, and fruit maturation. Other traits cannot be observed with an egg type because their differentiation corresponds to meristem's level, like floral initiation. Dotted lines refer to species for which there is a variable degree of overlapping among organs undergoing differentiation. Different scales have been developed based on the concept of describing the observable phenotype, like this one by figs for wheat, a grass species that grows along winter and spring. Let's check first the division among the three big phases of development, that is, the vegetative, the reproductive, and the maturation phases. Based on the visual phenotype, we can appreciate the relatively long duration of the vegetative phase, so described because only vegetative organs like leaves and stems can be observed. Next, we can appreciate the subsequent phases usually used for describing wheat life cycle. Crop emergence is followed by seedling growth. These phases are relatively short and followed by the tillering phase, during which secondary stems are produced from axillary buds located at the base of the main stem. Tillering is usually the longest phase in winter cereals. Stem elongation starts at the moment described as first node visible. The phase of stem elongation continues until spikes are visible, described as heading. Flowering takes place after heading. In wheat, flowering is described as the exposure of anthers, anthesis, that is pollen release, and ovaries fertilization have already taken place and are followed by grain filling until their physiological maturity. Harvest maturity is usually delayed until grain water content reaches the commercial standard. An important objective of scales was to unify crop description among scientists to facilitate comparisons across studies. Scales for the same crop, however, give variable detail within each phase, depending upon the objective of its developer. For instance, more detail given in early crop phases when aimed to most herbicides application, or in late phases for some insect control. After the development of scales for describing the external phenotype, focus was placed on meristem's activity through plant dissection. The so-called internal phenotype became evident, a step that improved our understanding of environmental effects on crops. Details were added to previously described stages, as the start and end of floral differentiation. The assessment of some phases, as the juvenile phase, required of specific experiments for its correct quantification. It is important to point out the distinction made between phenological events and phenological phases. The former are usually used for describing the milestones that identify the start and the end of each phase. Events are usually of a shorter duration than phases. For instance, we usually identify crop flowering date as an exact calendar date, say 1st October for wheat, simply because we describe flowering as the date when 50% of the visible spikes reach anthesis. Contrary, tillering does not take place on an exact date. It extends between the dates when 50% of the plants have at least one visible tiller to the date when 50% of the plants have started the stem elongation. Meristem's studies allowed for an improved distinction between vegetative and reproductive phases, a critical aspect for management decisions as herbicide application. Based on the meristems, we now realize that the vegetative phase is not as long as proposed by studies based on the external phenotype, though we still see plants in vegetative condition with tillers and leaves growing. Floral initiation has taken place in the apical meristem of the main stem, which has started differentiation of reproductive organs. Reproductive differentiation will continue even when stems start active growth at the jointing phase, which means an overlapping with active growth of spikes. The number of fertile florets will be defined at the end of the jointing phase, together with final length of stems and spikes. A large proportion of differentiated flowers will abort between the flag leaf stage and anthesis. All mentioned processes and events are evident only by plant dissection. Fertilized ovaries that succeed to set the grain will proceed to the next stage, the grain filling phase. Development of the new embryo starts at ovary fertilization and includes the differentiation of leaf and root primordia in the growing seeds, here identified as leaves initiation. The description made for wheat can be extended to all annual species produced for grain, as we can see in this example for sunflower, which is a composite with a terminal inflorescence like wheat. Once again, floral initiation takes place early in the cycle and sets a short duration of the actual vegetative phase respect to the actual reproductive phase. The expansion of leaves overlaps the periods of leaves initiation, flowers initiation, flowers development and growth. Flowering is represented by the anthesis period, which proceeds from the peripheral area of the head towards its center. In legume crops, here represented by soybean, the overlapping between leaf and floral primordia differentiation by meristems is more pronounced than in other crops. Meristems are distributed all around the plant, and for this reason we distinguish floral initiation from main shoot terminal apex change, which were equivalent among crops described previously. In legumes, floral initiation is the first initiated flower in an axial bud of a main stem leaf. A relevant aspect we must highlight for all evaluated crops is that organ development, represented by tissue differentiation, is markedly anticipated respect to organ growth, represented by its expansion and biomass accumulation. This fact is described in this schematic representation of the maize cycle. Considering leaves, the maximum number of leaf primordia differentiated by the apical meristem will be defined early in the cycle, at floral initiation in the terminal apex. In this example, at V6, that is when the plants have 6 visible leaves. However, we must wait the cycle to proceed up to flowering for complete leaf expansion of all differentiated leaves. In other words, for a maize plant with a total number of 18 leaves and floral initiation at V6, the expansion of leaves number 7 to number 18 takes place between V6 and anthesis. Let's analyze the last description, assuming plants growing at a constant temperature in a growth chamber. We will start considering leaf primordia in the apical meristem of a cereal plant. Firstly, there is a number of pre-established primordia in the seed embryo, which have been differentiated along seed formation in the mother plant. At sowing, after seed imbibition, the meristem will recover its biological activity and will continue differentiating leaves because it is in a vegetative stage. It will continue doing so until it changes from vegetative to reproductive at floral initiation, when it will start the differentiation of flower primordia. The total number of leaves at the given stem will be established at this moment and will never change. Because we are analyzing plants growing at a constant temperature, the rate of leaf primordia differentiation by the meristem is constant. This rate is described as the rate of leaf initiation and has units of number of primordia per day. The inverse of this rate, that is the number of days required for the differentiation of one leaf by the meristem, is described as the plastochron, from chronos, that means time, and plasto, that refers to the origin. Let's link meristem activity with visible above-ground plant activity, that is, with plants expanding their leaves. The first aspect is that the line representative of the number of visible leaves does not start at the given number of primordia. It starts at 0 at the time of crop emergence, which is delayed from sowing. We must recall that the first visible leaf of the crop will be the first leaf differentiated by the embryo when the seed was growing attached to the spike of the mother plant. At crop emergence, the meristem has already restarted its activity and is differentiating leaves. From crop emergence onwards, new leaves will be growing, expanding in the main stem. This process will take place at a constant rate for plants growing at a constant temperature. The expansion of leaves will continue until the last differentiated leaf has completed growth and becomes visible at the top of the plant soon before heading. This leaf is usually described as flag leaf in cereal crops and represents the last leaf differentiated by the meristem, that is, the youngest leaf of the plant. As for leaf primordia, the whole process from the first to the flag leaf is described by the rate of leaf appearance and has units of number of leaves per day. Its inverse is known as the phyllochron and has units of days per leaf. Important to highlight that floral initiation is not linked to any visible event in plants. As already explained, the reproductive period takes place from floral initiation to flowering and is far longer than the vegetative stage usually proposed by scales based in the external visible phenotype.